what would be? This percentage would be 2.5%. Correct? It is 95 up to here. Anything beyond 2 standard deviations will be 100 minus 95 will be 5%, but that is one side is 2.5, another side is 2.5. So any person who is having beyond 2 standard deviation which are above the mean, above 2 standard deviation above this mean, there will be only 2.5% in the population. That will be equal to your disease problem. Okay, that's not more than greater percentages. Okay, that's what I went by. So these values are important. So familial aggregation of quantity effects, how does that happen? So one way is to do the correlation among the relatives. So you need to do correlation, not, a correlation is not an association. A correlation is a mathematical term, an association is a qualitative term. Okay. So you need to draw the correlation graphs. Correlation is nothing but a graph which will show you if you have a height, and let us say your weight which is dependent on the height. Okay? So the height will give you a line like that. Okay? That is because, let us say you take multiple points. In statistics you won't get a curve like that. In statistics what you're going to get is the dots. Several dots here. Okay? If all these dots will show a pattern in this way, that means you're drawing a line like that, and that is called a positive correlation. That is, the line goes from left to right in that direction. It's a positive correlation. But sometimes, you have a question? So sometimes the curve can go from here to there. That is called a negative correlation. Okay? So those two correlations are there. For example, high blood pressure is there in all the relatives. That's a positive correlation. Sometimes negative correlation is possible. So this is average height and average height of the children. This is average height of the parents and average height of the children. That's what he plotted here. So you can predict that this is a positive correlation. Okay? You have all people who are all taller in a family, you are going to get taller children. That's what it is telling you. That means a similar correlation can be applied for a quantitative trait. So the quantitative trait is having in all parents, and that quantitative trait can also come in children. That is what the prediction. But you need to first establish this correlation curve, and correlation curve is essentially very similar to standard deviation. Similar type of formula is there, and you can use that formula to get these numbers. Okay? So now heritability. Heritability is a fraction of the total phenotype variance of the quantitative phenotype. So greater the heritability value, this is again just a simple mathematical value, greater the contribution of the genetic differences among the people in causing the variability of the trait. So this h square, I didn't put that 2 up there, the h square equal to 1, that is what the heritability value. If it is equal to 1, genes are totally responsible for phenotypic variance. So how do you estimate heritability from the twin studies? So you measure this variance in dizoid and digoid twins. That is, the variance means we just talked about the value of, instead of standard deviation, the variance is nothing but without the square root, that is s square is the variance. Okay? So that's what you will study. So h square, that is your variance, is variance in dizygotic pairs minus variance in monozygotic pairs divided by variance in dizygotic pairs. That is what the h square that you need to determine. That will give you the value, the heritability value. So again, there are various limitations of familial aggregation and concord heritability studies. Okay? So I'm not going to go through this, but there are some exceptions to the general rule. You need to know what the difficulties are. Let us give some examples of the disorders while the multifactorial inheritance is there. One is a cystic fibrosis. You know, cystic fibrosis is obviously a mucus problem we talked about. But you also can measure that as a quantitative trait. The quantitative trait is nothing but forced expiratory volume. That is, when you go to a clinic, I think they will ask you to blow in a small tube. How much of maximum that you can blow out. That's called forced expiratory volume. So, obviously, they have studied these forced expiratory volumes, the monozygotic and dizygotic twins, and they got a H square value of half, not 1. So what is this half is telling? That it is determined not by one locus, there are more than one locus in that particular gene. So H square tells you that there are more than modifier genes are there. If the quantitative trait were not to be discovered, let us say you are going by quantitative traits, qualitative trait by looking at his mucus. That's a qualitative trait. You are not really measuring the quantity. Then you would tell this fellow is cystic fibrosis, this fellow is cystic fibrosis, this fellow is cystic fibrosis. So your value would be H square will be equal to 1. But when you measure quantitative traits, what you are going to get is the different values of H square that will also tell you the relative amount. There is more than one locus. Therefore, you have to keep searching for other genes. Okay? In fact, they found 2 other genes like mannose binding protein as well as the TGF, that's beta. These 2 genes have been found to be involved in cystic fibrosis associations as well. So it's not purely that there's a channel protein in cystic fibrosis. In addition to that, the other 2 genes have been associated. Okay? And there are other multifactorial traits. For example, the retinitis pigmentosa is another disorder. So if you take a look at the retinitis pigmentosa, there is 2 genes are involved. The top, there is 1 gene, the bottom, there is 1 more gene. So in this particular fellow, he is a mutant for both genes. It's heterozygous all right, but both mutations are there. Whereas when you cross it and then just get segregation, if you get a plus and a plus mutant, there is no disease. So it has to be, both genes must be expressed. So look at here. So in this family, you are not getting for all individuals. In fact, all these individuals are now scot-free. That is because in first generation you got the disease, but because of segregation, again independent assortment, you are going to not get the disease. So how will you study a polygenetic disorder? If you go to your family, collect samples from the first generation, you got the disease. Second generation, you don't have. So how will you even want to pinpoint the gene where exactly it is located? That is where difficulties come in calculating these kind of disorders. Okay? So the only way that best measure is to get this kind of a rough estimates, whether H square is equal to 0.5, what is the familial aggregation numbers, what is the concordance and discordance values. Those are the things that you can study and tell, hey look, my genetic component is only less, genetic factors are more, environment. These are the only things that you can do at this stage of genetic scale. Okay? But maybe in the future, we may have better technologies. Another multifactorial disorder is the diseases or the diseases which we have been working for 30 years in our lab. Okay? So on this slide, I can lecture you for about 20 hours, so I'm not going to do that. What we study is thrombosis. Thrombosis is a highly complex disorder. That is because it is multifactorial, because actually somewhere in 1800s, Rudolf Virchow, who is the father of pathology, he pointed out there are 3 things can go wrong to cause thrombosis. But those 3 things since that time have been found to be more than 3, because he told a broad definition. What is thrombosis? Thrombosis is nothing but blood clotting inside the vessel. Blood clots inside the vessel. What is a blood vessel? Blood vessel is nothing but a tube containing cells which are lining the tube called endothelium, and there is a liquid which is flowing called plasma, and there are lots and lots of factors associated with the plasma. There is coagulation factors, etc. So there is tube, there are factors in that, cells and plasma. It is second component. The tube is the first component. The third component is the flow itself. The flow is determined by the heart. Heart pumps and the flow goes. So if the flow is very, very low, let us say there is what is it called, a stasis. If the flow is low, you're going to get more thrombosis. As long as it's constantly flowing, it is fine. if the plasmatic factors are there, a lot of them, okay, if some of them increase, let us say this increases, you're going to have this product more. That means the fibrin is a clot. That is much more. If this increases, that's going to produce more of this, that's going to convert more of that. This is called clotting cascade. Clotting cascade works very simply. There is an enzyme which cleaves one other protein, and that protein becomes an enzyme, and that enzyme cleaves another protein, and that becomes an enzyme, and so on. That is just nothing but a clotting cascade. So when you get injured, what happens is in the vessel, there is an endothelium. Beneath the endothelium, we call it subendothelium. When you puncture it, the subendothelium has got a lot of proteins that will get exposed. The platelets will come and sit on it and get stuck. It's called platelet aggregation. And then the clotting factors will come and sit on the surface of these platelets. And that is how these clotting factors are generating, and that is called a fibrin clot. So all these factors get activated and lead to a clot formation. And that clot, when it forms inside the vessel, it is thrombosis. When it is outside the vessel, it is called hemostasis, because that prevents you from bleeding, not homeostasis. Homeostasis is a different word. Hemostasis means blood, stasis means stopping. Okay? So this is a complex disorder. Now you've got all these factors have been figured out. A lot of platelet factors have been figured out. But flow factors, endothelial factors, nobody knows still. That's what we're working on. So these kind of disorders are called again multifactorial. Okay? There is one disease called factor V Leiden. This is very important. And also there is a prothrombin mutation. These two genes are extremely important in thrombosis. If a person is suffering from factor V Leiden mutation, factor V Leiden mutation means, I think you need to understand this cascade a little bit so that you can understand what is factor V Leiden. So this is a clotting cascade. This factor called factor 10A, don't worry about how it is generated, there's a big cascade, I'm not going to go through that. Factor 10A cleaves an inactive prothrombin, makes it thrombin, and thrombin converts fibrinogen to fibrin. So until you get injured, all these factors are floating in the blood as inactive forms. Only when you are injured, they get activated. Otherwise, your whole system will be completely clogged, right? That's not what we want. But there is also a factor called factor 5, which gets converted to factor 5A by feedback cleavage by thrombin. And this factor 5A is a cofactor for factor 10A. Okay? So if you have a mutation here that's called factor V Leiden, okay? So this particular mutation will not let this factor 5A degrade completely. So that mutation is causing stability of that whatever that factor is. Therefore, it continuously keeps converting or activates factor 10A continuously and generates more and more clot. That means that factor V Leiden mutation is going to create more clot than less clot. That's why it is called thrombosis. Whereas in another mutation called prothrombin, this particular mutation is not at all located in the coding sequence. It is entirely in the non-coding sequence of the mRNA. So non-coding sequence of mRNA means it could be either UTR or 5' UTR, right? What does it do? It creates the stability of the messenger RNA. That mutation makes this messenger RNA more stable. More stable messenger RNA means less degradation, right? Less degradation means there is more amount of protein production. That is, more prothrombin is produced. If more prothrombin is produced, you are going to have more prothrombin here. That 10A can convert more prothrombin to more thrombin and more fibrin formation. So either way, these two mutations have been very, very important in families which are having thrombosis. So if you have, a lot of them are having these kind of mutations in the families. But there are a lot of variations in this game. Okay, I'm not going to go through the details of that. But if you have this clot in your heart vessel supplying to the heart, you call it myocardial infarction. If you have a clot in your veins inside the legs, you call it deep vein thrombosis. If you have a clot in the brain, you call it